Hello and welcome to the channel. It seems that the cloudy sky is beginning to clear for patients with Alzheimer's disease and their families. Since its first description in 1906, Alzheimer's disease has been without an effective cure, with treatments limited to relieving symptoms. I recently discussed the controversial approval of Biogen's new drug, aducanumab, for Alzheimer's disease, as you can see now somewhere on your screen. Now, it seems that an additional drug for Alzheimer's disease is nearing approval by the FDA, and more are on the near horizon as well. But what is the true difference between these new treatments for Alzheimer's disease, and how effective are they expected to be? That will be the topic of today's video. The pharmaceutical company Biogen was given approval by the FDA for aducanumab, a drug that targets amyloid-beta, one of the proteins known to accumulate in the brains of patients with Alzheimer's disease. But which portion of the amyloid-beta protein does it target, and is it any different from other drugs that are currently under different stages of development? Key to understand the difference between current and future therapies for Alzheimer's disease is to know that most scientists think that the combined effect of the accumulation of two specific proteins in the brain is critical for the initiation of Alzheimer's disease. Specifically, one of the proteins is called amyloid-beta, and it accumulates outside of the cells, in structures called amyloid plaques. The second protein accumulates within the cells, or intracellularly, and is called tau, in structures known as neurofibrillary tangles. What exactly causes the accumulation of these two proteins in the brain in the first place? That's a matter of scientific debate and a topic for a different video. But for now, it is sufficient to say that all the drugs we discuss here today target one of these two proteins. But let us start with Biogen's drug, aducanumab, that was recently approved for use by the FDA. Aducanumab is in fact an antibody designed to bind amino acids 3 to 7 of the amyloid-beta peptide. The basis for its effect is its ability to bind amyloid-beta, labeling it for microglial cells that later arrive and phagocytose, or in other words, swallow the toxic protein. This drug was approved by the FDA in what some scientists called a highly controversial move. It isn't very clear yet who exactly will be able to get this treatment and how early will it be possible to initiate treatment. But this doesn't end there. Eli Lilly, another US-based pharmaceutical company, develops too a drug which is also an antibody called donanemab that targets amyloid-beta. But this time, this antibody doesn't simply bind to a specific amino acid sequence on the protein, but it rather binds a distinct form of amyloid-beta that is different from the one that Biogen's aducanumab is targeting. Specifically, it is truncated in its N-terminal region, as well as chemically modified. This modification, called pyroglutamate, is thought to render amyloid-beta more hydrophobic, which causes it to be better at repelling water molecules and as a result aggregate more than a typical amyloid-beta peptide. Aggregated amyloid-beta forms plaques, which suggests that perhaps Eli Lilly's antibody donanemab will be mostly targeting the affected brain regions rather than spread all over the brain. So why do I mention Eli Lilly's donanemab anyways? Well, apparently Eli Lilly's donanemab was recently granted an FDA breakthrough therapy status. As a result, Eli Lilly intends to submit a biologics license application, or BLA in short, to the FDA later on this year. This would actually allow Eli Lilly to market donanemab in the US. But what is a breakthrough therapy designation anyways? So the FDA's breakthrough therapy designation is intended to speed the development and review of treatments for serious medical conditions. To receive such status, candidate therapies must demonstrate significant improvement over already existing FDA-approved treatments. This decision by the FDA was based on results from a phase 2 clinical trial called TRAILBLAZER-ALZ that evaluated donanemab's safety, tolerability, and efficacy in adults with early-stage symptomatic Alzheimer's disease. In addition to the TRAILBLAZER-ALZ clinical trial, 2 other clinical trials are currently assessing the safety and efficacy of donanemab for patients with early Alzheimer's disease. All this suggests that soon, perhaps within a year, Alzheimer's disease patients will have two optional treatments that target amyloid-beta pathology in their brains. But amyloid-beta is not the only druggable target in Alzheimer's disease. Many scientists think that the tau protein is a much better target for treating Alzheimer's disease. Some scientists actually use quite harsh language when relating to drugs that target amyloid-beta. For example, and I quote, "Clinical trials with these agents should be abandoned and more time and money spent on further investigating the underlying cause of Alzheimer's disease." Indeed, some companies develop antibodies that target tau and test them in clinical trials. Tau is a cytosolic protein that typically stabilizes tubulin filaments which help regulate intracellular transport. For many different reasons, if tau undergoes multiple phosphorylation events, it is no longer able to stabilize tubulin in the cell, and two things happen next. One, tubulin starts to disintegrate, and second, tau begins to aggregate. The combined effect of these two events is neuronal dysregulation and cell death, which is at the root of cognitive decline in patients with Alzheimer's disease. And therefore, although characterized as a cytosolic protein, tau is reported to be found extracellularly under normal physiological conditions in the brain interstitial fluid or ISF, in the cerebrospinal fluid or CSF, and plasma of rodents, non-human primates, as well as humans. Although the mechanism of tau excretion from the cells is not fully understood, it is hypothesized that in the context of Alzheimer's disease and possibly other tauopathies, diseases that involve the protein tau such as frontal dementia, pathogenic tau species spread from one cell to another via the extracellular environment. As I mentioned, several pharmaceutical companies are currently developing and testing drugs and specifically antibodies against tau as a potential therapy for tauopathies, including Alzheimer's disease. The first antibodies that were developed against tau targeted its N-terminus because it produced the highest affinity antibodies. However, it seems that none of the antibodies that bind this region of the tau protein have had any success in phase 2 clinical trials to date. Both Biogen and AbbVie posted negative findings following testing their tau-directed antibodies for diseases called progressive supranuclear palsy, which is characterized by tau pathology. Their clinical trial have been discontinued for this disorder but remains in trials for Alzheimer's disease at present. In another example, Roche developed a humanized anti-tau IgG4 monoclonal antibody that binds the N-terminus of all 6 human tau forms, both monomeric and oligomeric forms, regardless of its phosphorylation status. In a placebo-controlled clinical trial, results were quite disappointing. In fact, despite treatment with this antibody, there was no change in any clinical endpoint, and the antibody did not slow tangle accumulation in the brain relative to placebo. In fact, non-invasive imaging for tau using PET or positron emission tomography showed a significant increase in tau accumulation. As I mentioned, these 3 drugs targeted the N-terminal region of tau, and by now it can be concluded that antibodies that target this region of tau are ineffective in changing the course of the disease. As a result, additional drugs are currently developed for different regions within the tau protein, and it may take several years until positive news will emerge from these efforts. In summary, amyloid-centered treatments currently dominate the therapeutic arena for Alzheimer's disease patients, while tau-centered therapies are yet to mature. In the near future, I will make a more in-depth discussion of the different druggable regions on the tau protein, as significant efforts are currently made in this field. This will be all for today. You're welcome to continue the discussion in the comments below. Until the next video, stay safe.